This figure illustrates how rituximab, a therapeutic monoclonal antibody, targets and destroys B cells expressing CD20 surface antigen. The diagram breaks down this process into three primary therapeutic mechanisms: antibody-dependent cellular cytotoxicity, along with phagocytosis, complement-dependent cytotoxicity, and direct induction of apoptosis. Here is a detailed breakdown of each of the mechanism as depicted in this figure. First one, antibody-dependent cellular cytotoxicity and phagocytosis. The diagram highlights how rituximab acts as a molecular bridge between the target B cell and the effector immune cells. The variable region, orange Y shape of the rituximab antibody binds specifically to CD20 antigen, yellow triangles, on the B cell membrane, while the constant region Fc portion of the antibody binds to Fc gamma receptor, red blocks expressed on the effector cells. Natural killer cells activation. When NK cell, the purple cells, bind to the rituximab coated B cell via its Fc gamma receptor, it triggers the release of cytotoxins, represented by green dots. These perforins and granzymes induce targeted cell lysis of the B cell. Macrophage phagocytosis. Similarly, macrophages, the pink cells, utilize their Fc gamma receptor to recognize the rituximab bound B cell, initiating phagocytosis to engulf and destroy the target cell. Second pathway, complement dependent cytotoxicity. The upper right portion of the diagram shows classical complement pathway activation when multiple rituximab antibodies bind to CD20 molecules closely clustered on the B cell surface. They bind to and activate the complement proteins circulating in the blood, represented by the light green squares. This cascade culminates in the assembly of membrane attack complex, a pore-like structure labeled as green block that inserts itself directly into the B cell membrane. The MAC disrupts the cell's osmotic balance, leading to cell death and lysis. Third mechanism, direct induction of apoptosis. In addition to recruiting external immune system components, the binding of rituximab to CD20 can trigger intracellular signals directly. As shown on the left side of the B cell, the cross-linking of CD20 by rituximab can directly initiate apoptosis from within the B cell itself, independent of complement proteins or effector immune system. So the summary of the image is CD20 yellow triangles, the specific target protein marker found on B cells. Rituximab orange Y, the monoclonal antibody drug targeting CD20. Fc gamma receptor red C shape is the receptor on immune cells, macrophage and NK cells that locks onto the antibody. MAC stands for membrane attack complex that punctures the target cell membrane during the complement activation.